Welcome back, my dear students. Today we will explore the fascinating world of the invertebrate endocrine system and physiology, the hidden chemical control network in creatures like insects and crustaceans. To help you grasp this complex topic more easily, I have referred to a series of standard textbooks and research articles to prepare this presentation, ensuring that you get reliable, well structured and exam relevant information for your better understanding and retention. We will learn about insect hormones, their types and the special glands that release them, as well as how these hormones regulate their growth, metamorphosis, molting and diapause. We will also discover the vertebrate type hormones found in crustaceans and the roles of the X organ, Y organ and the other neurochemical organs. So get ready to see how tiny hormones bring about big changes in the lives of these amazing animals. Let's get started. Introduction. Endocrine system in invertebrates is a collection of neurosecretory cells and endocrine glands that regulate physiology and behavior via hormones. It controls growth, development, reproduction, metabolism and behavior. Unlike vertebrates, it lacks a complex network of ductless glands, and most hormones are produced directly by specialized nerve cells. In invertebrates, across different phyla, from simple nerve nets in lower forms to specialized glands in advanced groups, hormones are produced by neurosecretory cells and other structures to control growth, reproduction, molting, regeneration and other vital functions. Now we should study endocrine regulation across different invertebrate phyla, their structure, hormones and functions. First of all, we see about the cnidarians, which include Hydra, Aurelia. Their simple nerve net contains neurosecretory cells that release factors promoting growth and development. Even these primitive creatures use hormone to regenerate lost parts. In case of Ctenophora or comb jellies with their glowing ciliated bodies, comb jellies use neurosecretory cells in nerve net and arboreal organ to control their ciliary breathing and locomotion and reproduction through peptide-like messengers. Platyhelminthes, or in case of flatworms, which are famous for regeneration, these worms have neurosecretory cells in their cerebral ganglia and ventral nerve cords. Their hormones control their growth, reproduction and the process of regeneration. Parasitic flatworms even release substances to manipulate their host's physiology. Nemathelminthes, or roundworm, their nerve ring and longitudinal ganglia release ecdysteroid-like hormones for molting, growth and reproduction. Special sense organs like amphids and phasmids may also have neurosecretory roles. This is nemathelminthes or roundworms. Annelids, earthworms, polychaetes and leeches. In earthworms, the cerebral ganglia release hormones for regeneration, growth and reproduction. In polychaetes, a caudal neurosecretory system regulates spawning, especially during breeding seasons. Arthropods, in case of crustaceans, in crabs or shrimps, their X-organ sinus gland or the eye stalk, they release molting inhibiting hormone or MIH and pigment hormones. Y organ secrete ecdysteroids to trigger molting. Androgenic gland determines male characteristics. Mandibular organ produces methyl farnesate, similar to juvenile hormone in insects. In case of insects like cockroach and butterfly, their neurosecretory cells in the brain release PTTH or prothoracicotropic hormone. Corpora cardiaca, they store and release hormones for metabolism and heart rate. Their corpora alata produce juvenile hormone, which is crucial for metamorphosis and reproduction. Prothoracic glands, they produce exudation to initiate molting. Mollusk, or in case of snails, clams and octopuses. In cephalopods like octopus, the optic gland controls gonad maturation and reproduction. Other ganglia, cerebral, pleural, pedal, visceral, they secrete hormones for growth, spawning and osmoregulation. Their echinoderms include starfish, sea urchins. Their radial nerve cords and nerve ring have neurosecretory cells that regulate regeneration, pigment movement, and reproductive timing. Hemichordata. In hemichordates like Balanoglossus, the collar cord contains neurosecretory cells that control growth and seasonal breeding patterns, bringing them closer to the chordates in hormonal complexity. This is their dorsal nerve cord. Acha, insects hormone. Now, the types and the release sites. Brain neurosecretory hormones. Their source is neurosecretory cells in the brain in their protocerebrum. The key hormone is protothoracic hormone or PTTH, which stimulates prothoracic glands to secrete ecdysone. Their release sites include corpora cardiaca, which stores and releases PTTH into hemolymph. Corpora cardiaca hormones, their source is the paired glands behind the brain. They store and release PTTH from brain. Cardio-acceleratory hormones increase heart rate. Adipokinetin hormones mobilizing energy reserves. Release site, it directly release into the hemolymph. Corpora allata hormones, the small endocrine glands near corpora cardiaca. Key hormone, juvenile hormone, maintains larval state, regulates metamorphosis and reproduction. Release site, they directly move into their hemolymph. Prothoracic gland hormones, source prothoracic glands in thorax. Key hormone, ecdysone or molting hormone triggers molting and metamorphosis. Release site into hemolymph, converted to active form like 20-hydroxyecdysone in peripheral tissues. Other hormones, eclosion hormone from brain and ventral nerve cord ganglia initiates emergence from pupal cuticle. Bursicon from neurosecretory cells hardens and darkens new cuticle after molting. Diuretic hormones from brain and thoracic ganglia regulate water balance. Endocrine regulation of insect growth, metamorphosis, molting and diapause. So what's the introduction? Firstly, insects have a neuroendocrine system combining neurosecretory cells and endocrine glands. Growth, molting, metamorphosis and diapause are regulated by a coordinated action of three major endocrine centers. Number one, neurosecretory cells of brain or prothoracicotropic hormone or PTTH, prothoracic glands which secrete ecdysone, and corpora allata which secretes juvenile hormone. Endocrine regulation of growth and molting, which is ecdysis, growth and stretch receptors. When the larva grows and cuticle becomes tight, stretch receptors in their body wall signals the brain. Their brain activation, neurosecretory cells in the brain release PTTH into the corpora cardiaca, released into their hemolymph through their open circulation. Ecdysone secretion, PTTH hormone stimulates prothoracic glands to secrete ecdysone. Ecdysone activation, ecdysone is converted to peripheral tissues to 20-hydroxyecdysone, which is the active form. Cuticle separation or apolysis, 20-hydroxyecdysone triggers separation of old cuticle from the epidermis, cuticle digestion and new cuticle formation. Epidermal cells secrete molting fluid and synthesize new cuticle beneath old one. Ecdysis or shedding, eclosion hormone initiates shedding behavior. Bursicon hardens and darkens the new cuticle. Endocrine control of metamorphosis. Role of juvenile hormone. Their high juvenile hormone with ecdysone promote larval molt and retains the larval features. But low juvenile hormone with the ecdysone, it promote larval pupal molt and it is the transition between different larval stages. But if you apply no juvenile hormone but you apply ecdysone, then pupa adult mole will occur, which is the final term of metamorphosis. So we should know what are the different types of metamorphosis. There are three types. Ametabolous, there is no metamorphosis but molting continues into the adulthood. It is found in case of silverfish or bookworm. Hemimetabolous, their gradual metamorphosis occurs. The nymphs resemble adults. It is found in case of grasshopper. And in case of holometabolous, complete metamorphosis is found and the larva is converted into the pupa and subsequently into adult, which is found in case of butterfly. Now we should study endocrine regulation of diapause. Diapause is basically a hormone controlled developmental arrest to survive adverse conditions in case of, for example, winter drought. Regulation, it controlled mainly by juvenile hormone and environmental cues like photoperiod, temperature, etc. In many species, corpora alata suppression uh, deaccelerate the juvenile hormone production and diapause is initiated. But in case of reactivation of corpora alata, juvenile hormone is promoted and then diapause is terminated. So in case of Bombyx mori or silkworm, photoperiod affects the brain, alters PTDH release and changes juvenile hormone secretion, which induces or ends the process of diapause. Now, we know vertebrate type hormones. In case of crustaceans, they are usually X organ, Y organ and associated neurochemical organs. Crustaceans like crabs, lobster, prawn, these are their image, possess a well-developed neuroendocrine system that integrates nervous and endocrine functions similar in principle to vertebrates. So the term vertebrate type hormones specifically refers to chemical messenger in crustaceans that are analogous in structure or function to those found in vertebrates, including steroids, peptides, amines, etc. So the found key endocrine centers found in crustaceans are the X organ sinus gland complex, Y organs and other neurosecretory or neurohemal structures. First of all, we know the X organ sinus gland complex. They are found in the medulla terminalis of their eye stalk. It is the cluster of neurosecretory cells, the neurohemal organ that store and release hormones from the X organ into the hemolymph. The hormone produced from this structure include molt inhibiting hormone or MIH, which inhibits Y organ activity, preventing ecdysone synthesis until the appropriate time. Crustaceans hyperglycemic hormone or CHH, which regulates blood glucose level analogous to vertebrate glucagon. Gonad inhibiting hormone or GIH delays gonadal maturation, parallels certain vertebrate gonadotropin regulating hormone. Mandibular organ inhibiting hormone or MOIH inhibits mandibular organ function affecting methyl farnesate synthesis. Pigment concentrating or dispersing hormones regulate chromatophore pigment migration for color change, functionally comparable to melanocyte stimulating hormone or MSH in vertebrates. Functions include regulate molting cycle, control reproduction, maintains energy homeostasis, modulates color change for camouflage or signaling. This is the image showing the X organ and this image of the prawn showing the presence of the Y organ. Y organ are basically paired glands located in the maxillary or antennal segments. Their function is the primary site for ecdysteroid or ecdysone synthesis. Molting hormones structurally similar to vertebrate steroid hormones. They are inhibited by MIH from X organ sinus gland complex. When they are activated, when MIH secretion declines, they are often due to environmental cues like photoperiod, temperature and growth stage. The hormonal product include ecdysone, which is converted to 20-hydroxyecdysone in peripheral tissues. It controls apolysis, cuticle synthesis and ecdysis. Associated neurochemical organs. Besides the X and Y organs, crustaceans have other vertebrate hormone releasing structures like pericardial organs. They are located on the lateral walls of the pericardial cavity. Its function is the release of cardioactive amines. They release serotonin, dopamine, octopamine, influencing heart rate, muscle contraction, etc. They are androgenic glands located base of the fifth pre-opioid in mammals. Their function is to secrete androgenic hormones controlling development of male sexual characteristics, which is analogous to vertebrate androgen. Mandibular organs at the base of the mandibles. They structurally related to insect juvenile hormone. They regulate reproduction, growth and metamorphosis. The regulation is inhibited by MOIH from X organ sinus gland complex. commissural ganglia that contain neurosecretory cells producing hormones for gut motility and digestion, like vertebrate-like gastrointestinal peptides. So, I hope this class helped you understand invertebrate endocrine system and physiology, how insect and crustacean hormones are produced, their release sites and their vital roles in growth, metamorphosis, molting and diapause. So stay curious about how these tiny chemical messengers control big biological changes. If this helped, please like, share and subscribe. See you in the next class. Thank you.